Ever wondered what makes a tiny bacterium so fundamentally different from, say, your own cells? And why those microscopic differences actually matter for things like antibiotics or, how infections even spread? Today, we're taking a deep dive into exactly that. We're exploring the incredible, often unseen world of life's fundamental architecture, prokaryotic and eukaryotic cells. We've pulled insights directly from a really fantastic source, a chapter from Microbiology, an introduction, the 13th edition. Our mission for you today is to distill the most important nuggets, unpack some complex ideas, basically give you a shortcut to understanding the incredible diversity and, frankly, the cleverness of cells, from their basic building blocks right up to their surprising adaptations.
Yeah, and what's truly fascinating here is how these tiny differences in how cells are built can have massive implications. We're talking about everything from how a specific bacterium causes disease to how certain antibiotics actually work, and even touching on the very origins of complex life as we know it. We'll try to connect the dots between the big concepts, the microbial processes, specific structures, and even some real-world applications, highlighting, the clinical and environmental relevance as we go through this deep dive.
Okay, let's unpack this foundational idea right at the start then. All living cells fall into one of just two broad categories. So what's the big picture, the defining difference between prokaryotes and eukaryotes?
The main feature, the one that really distinguishes them, is the nucleus. Simple as that almost. Prokaryotes, which include bacteria and archaea, are prenucleus organisms. Their genetic material, their DNA, it's typically a single circular chromosome, and crucially, it's not enclosed by a membrane. It's just sort of there in the cytoplasm. Eukaryotes, on the other hand, that's us, plants, animals, fungi, protozoa, algae. We have what's called the true nucleus. Our DNA is organized into multiple linear chromosomes, and it's separated from the rest of the cell, the cytoplasm, by a distinct nuclear membrane.
Okay, so no membrane-bound nucleus for prokaryotes. What's the immediate implication of a cell not having one for like how it lives and reproduces?
Without that central compartment, their cellular processes are often much more streamlined, you could say, maybe simpler in some ways. Prokaryotes generally lack other specialized membrane-enclosed compartments, the organelles, like mitochondria or chloroplasts that eukaryotes have. Their DNA also isn't associated with histones. Those are the special proteins that help package DNA in eukaryotes. And when they divide, it's usually by a process called binary fission. It's a simpler way of splitting in two compared to the more elaborate dance of eikosis we see in eukaryotes.
It's quite remarkable, isn't it? Despite such fundamental structural differences, the core machinery of life seems conserved. They share some basic life processes with their cells, right?
Oh, absolutely. That's a key point. Both types of cells contain the same fundamental building blocks: nucleic acids, proteins, lipids, carbohydrates. The basics are there. And they use the same core chemical reactions to metabolize food, build protein, store energy. So the basic chemistry of life is very much shared. The major differences, as you said, are primarily structural, especially around things like their cell walls and the specific type of ribosomes they use to make proteins.
Right.
So with that overview, let's journey into the microscopic world of prokaryotes then. Specifically bacteria, this vast group of tiny unicellular organisms that are literally everywhere, inside us, around us. They might be small, but they're incredibly diverse. Incredibly diverse, yes.
Yeah.
Bacteria are differentiated by, loads of factors. Their shape, their chemical makeup, what they eat, their biochemical activities, where they get energy. And it's important to remember most bacteria out in nature aren't just floating around freely. They're usually found sticking to surfaces, often forming these complex communities we call biofilms. That's their preferred lifestyle, really.
Yeah.
Biofilms, right. We hear a lot about those. But okay, let's picture one. What do these tiny powerhouses actually look like under a microscope? What's their typical form?
They are remarkably small. Most bacteria are typically between, say, zero 0.2 to 2.0 micrometers in diameter and maybe 2 to 8 micrometers long. Tiny. And shape-wise, they generally come in three basic forms: spherical, which we call a coccus or cocci for plural. Then there's rod-shaped, or bacillus. And finally, spiral shapes, which includes things like fimbriae, spirilla, and spirochaetes, different kinds of twists and curves.
And they don't just exist as single cells. They often arrange themselves in distinct patterns, too, which actually helps in identifying them in the lab. Right?
Precisely. Yeah, that arrangement is often a clue.
Yeah.
For instance, cocci, the spherical ones, can form pairs, diplococci, or chains like beads on a string, streptococci, or even cube-like groups or grape-like clusters, staphylococci. You see those names in clinical contexts often. Bacilli, the rods, typically appear as single rods or maybe pairs or chains, the streptobacilli.
Oh, okay. And you mentioned bacilluses versus bacillus. Quick clarification there.
Good point. Yeah, it's a bit confusing. Bacillus, with a lowercase b just refers to that rod shape. Any rod-shaped bacterium is a bacillus, but bacillus with a capital B and italicized, that refers to a specific genus of bacteria, like Bacillus anthracis, the one that causes anthrax. So shape versus name.
Got it. Okay, let's move beyond just the shape. What's on the outside of these cells? What helps them survive and interact with their environment, which sometimes includes our bodies?
Well, many prokaryotes secrete this sticky, gelatinous layer on their surface. It's often called a sugar coat, technically a glycocalyx. Now, if this coat is well organized and firmly attached to the cell wall underneath, we call it capsule. If it's more unorganized, kind of loose and slimy, we refer to it as a slime layer. Same basic idea, just different consistency.
Right, a capsule or a slime layer. This glycocalyx seems like such a simple addition, maybe, but it has profound implications for human health, doesn't it? I mean, that's where virulence comes in.
It absolutely does. Capsules are often crucial for bacterial virulence. That's their ability to cause disease. They frequently protect pathogenic bacteria from being eaten, basically, by our immune cells, a process called phagocytosis. For instance, streptococcus pneumonia, a common cause of pneumonia, uses its capsule to evade our immune system. Without the capsule, it's much less dangerous. And the glycocalyx, particularly slime layers, is also vital for bacteria sticking to surfaces and forming those biofilms we mentioned earlier. There's something called an extracellular polymeric substance, or EPS. It's a type of glycocalyx that helps cells in a biofilm attach to their surroundings and to each other. This protects them, helps them communicate, and lets them survive on all sorts of surfaces, rocks, teeth, even medical implants like catheters. Big problem in hospitals. Vibrio cholerae, the bacterium causing cholera, uses its glycocalyx to attach to the small intestine. It also offers really vital protection against drying out.
So we've covered sticking and protection. What about getting around? How do bacteria actually move themselves? They're not just passive, right?
Not at all. Many bacteria use flagella. These are long, filamentous appendages that propel them through liquid environments. And interestingly, unlike the flagella in our cells, bacterial flagella rotate. They spin like tiny propellers. It's quite amazing engineering. Bacteria without flagella are called atricious. And the way flagella are arranged all over the cell, or just at one end or both ends, that also helps classify them.
That rotation sounds pretty complex for such a tiny structure. How does that work?
It is quite ingenious. Each flagellum is this incredibly complex molecular machine. It has a filament, a hook, and a basal body that anchors it and acts as the motor, using proton flow. Gram-negative bacteria even have a more complex basal body structure than gram-positives. This rotation allows bacteria to move in relatively straight lines, which we call runs. These runs are then interrupted by these random changes in direction known as tumbles. So it's like run, tumble, run, tumble.
Okay, run tumble. This raises an important question, though. Why do they tumble? Seems random, but is it?
It seems random, but it's actually part of a very sophisticated guidance system. They use these movements for taxis. That is directed movement toward or away from a stimulus. So if they sense they're moving towards something good, like a chemical attractant that's chemotaxis, or maybe light for photosynthetic bacteria, phototaxis, they'll suppress the tumbling. Lots of runs, fewer tumbles, heading in the right direction. But if they sense they're moving away from an attractant or towards something bad, a repellent, the frequency of tumbles increases, helping them reorient randomly until they find a better path. It's biased random walk. And the protein that makes up the flagellar filament, called H-antigen, is even used in labs to distinguish different strains within a bacterial species, like in E. coli O157H87.
Fascinating. And archaea, the other prokaryotes, they have something similar.
Yes, modal archaea have structures called archaea. They look similar, and they also rotate like flagella, but chemically they're different, made of proteins called archaelins, and they use ATP directly for energy, unlike bacterial flagella.
Okay. So beyond flagella and archaea, what other structures help bacteria interact with surfaces or maybe even other cells?
Well, there are axial filaments. These are unique to a group called spirochetes, which includes bacteria like Treponema pallidum, the cause of syphilis, and Borrelia burgdorferi, which causes Lyme disease. These axial filaments are bundles of fibers that lie beneath an outer sheath and wrap around the cell. When they rotate, they cause the whole cell to move in a corkscrew motion, which is great for moving through viscous fluids like mucus or tissues. Then you also have fimbriae and pili. These are usually shorter and straighter than flagella. Fimbriae are like little bristles, and their main job is adherence. They help bacteria stick to surfaces and to each other, which is absolutely crucial for forming biofilms and colonizing host tissues. For instance, Neisseria gonorrhoeae uses its fimbrio to attach to mucous membranes. Without them, it can't cause gonorrhea.
And pili, they do more than just attach, right? I remember reading about DNA transfer.
That's right. Piles are typically longer than fimbriae, and usually there are only one or two per cell. They're involved in a couple of types of twitching motility, kind of like using a grappling hook, and gliding motility. But the most famous role for some pili is conjugation. These are often called sex pili. They form a bridge between two bacterial cells, allowing for the transfer of DNA, usually plasmid DNA, from one cell to another. This process, called conjugation, is incredibly significant medically because it's a major way that genes for things like antibiotic resistance or new metabolic capabilities can spread rapidly through a bacterial population.
Wow, okay, so that's a lot going on outside the main cell body. Let's move inward. What about the main protective layer beneath all those external structures, the cell wall?
The cell wall. It's a really critical structure. It's complex, it's semi-rigid, and it does two main jobs. It gives the bacterial cell its characteristic shape, and maybe even more importantly, it prevents the cell from rupturing due to the high internal water pressure, osmotic lysis. It acts as a strong protective barrier. And clinically, it's a prime target for many antibiotics. Why? Because it's made of chemicals, primarily peptidoglycan, that are unique to bacteria and aren't found in human cells. Target the wall, kill the bacteria, leave the host unharmed. That's the goal.
Peptidoglycan, what exactly is that?
Peptidoglycan is this huge mesh-like molecule. Think of it like a chain-link fence wrapped around the cell, but in 3D. It's made of repeating sugar units, specifically NAG and NAM, linked together by short chains of amino acids, polypeptides. This creates a strong but somewhat flexible lattice structure that provides that essential structural support and protection. It's unique to bacteria.
And this unique structure, peptidoglycan, is precisely where the Gram stain comes in, right? Differentiating bacteria based on how much peptidoglycan they have and where it is.
Exactly. The Gram stain, developed over a century ago, is still fundamental in microbiology, and it works because of fundamental differences in the cell wall structure between what we call gram-positive and gram-negative bacteria. Gram-positive cell walls have many, many layers of peptidoglycan, forming a thick, rigid structure right on the outside. They also contain molecules called teichoic acids embedded within this thick layer. Because the peptidoglycan layer is so thick, it traps the crystal violet iodine complex used in the first step of the stain, even when you try to wash it out with alcohol. So they end up looking purple under the microscope. Gram-negative cell walls, on the other hand, have only a thin layer of peptidoglycan. And importantly, this thin layer is sandwiched between the inner plasma membrane and a unique outer membrane. This outer membrane is composed of lipopolysaccharides, LPS, lipoproteins, and phospholipids. During the Gram stain, the alcohol decolorizer dissolves this outer membrane and damages the thin peptidoglycan layer, allowing the crystal violet iodine complex to wash out easily. Then, when the counter stain, usually saffron, is applied, these decolorized cells pick it up and appear pink or red.
This outer membrane of gram-negative bacteria sounds like a pretty big deal then, not just for the Gram stain, but for how the bacteria interacts with the world and with us.
It absolutely is a big deal. That outer membrane provides a strong negative charge, which helps the bacteria evade phagocytosis by our immune cells. It also acts as a significant barrier. It blocks entry of many harmful substances, detergents, heavy metals, bile salts found in our gut, and crucially, many antibiotics, including penicillin, which can't easily cross it. Nutrients have to get in through specialized protein channels embedded in this outer membrane called porins.
And you mentioned lipopolysaccharide, LPS, in that outer membrane. I remember the clinical case notes mentioning it in relation to severe symptoms.
Yes, the lipopolysaccharide, LPS component, is particularly critical from a medical standpoint. It has three parts, but the key one is lipid A. Lipid A is buried in the outer membrane, and it acts as an endotoxin. When gram-negative bacteria die and break apart, lipid A is released, and it can trigger a powerful inflammatory response in the host. This is responsible for many of the severe symptoms associated with gram-negative bacterial infections, like high fever, dilation of blood vessels leading to shock, and problems with blood clotting. This is what can make infections from bacteria like Klebsiella pneumonia so dangerous. The other part, the O polysaccharide, extends outward and acts as an antigen, which our immune system can recognize. It's variable between strains, so it's useful for identification, similar to how teichoic acids are used in gram-positives.
Okay, so that's the gram-positive, gram-negative divide. But you hear about some bacteria that don't fit neatly into those categories because they have atypical cell walls. What are those exceptions?
Right, there are a few important exceptions. First, you have the mycoplasmas. These bacteria are unique because they naturally lack cell walls altogether. They just don't make peptidoglycan. This makes them naturally resistant to antibiotics like penicillin that target cell wall synthesis. To survive without a wall, their plasma membranes contain sterols, similar to eukaryotic cells, which provide some rigidity and protection from lysis. Second, archaea, which are prokaryotes but distinct from bacteria, have cell walls, but they don't contain peptidoglycan. Instead, they often have a substance called pseudomurine or other complex polymers. If you gram stain them, they usually appear gram-negative, but it's not because they have an outer membrane like gram-negative bacteria. And third, you have the acid-fast cell walls. These are characteristic of bacteria in the genera Mycobacterium, like the one causing tuberculosis, and Nocardia. They have a relatively thin peptidoglycan layer, but outside of that, they have a thick, waxy layer rich in lipids called mycolic acids. This waxy layer prevents the uptake of dyes used in the gram stain, so they don't stain well. They require a special staining procedure, the acid-fast stain, to be visualized. That waxy layer also makes them resistant to many disinfectants and antibiotics.
So what happens if these cell walls, particularly the peptidoglycan layer, get damaged? By enzymes or antibiotics?
Good question. Our own bodies produce an enzyme called lysozyme found in tears, saliva, and mucus. Lysozyme specifically breaks the bonds between the sugar units in peptidoglycan. If you treat a gram-positive cell with lysozyme, you can completely remove its cell wall, leaving behind just the plasma membrane enclosing the cytoplasm. This wallless cell is called a protoplast. If you do the same to a gram-negative cell, lysozyme damages the peptidoglycan, but the outer membrane often remains mostly intact. This results in a spheroplast. Both protoplasts and spheroplasts are very sensitive to osmotic changes. If they're in a solution with low solute concentration, hypotonic, water will rush in and they'll burst or lyse. Some bacteria can even spontaneously lose their cell walls under certain conditions and survive as L forms. These can sometimes persist during antibiotic therapy and then regrow their walls later, causing relapses.
And that brings us right back to antibiotics like penicillin. Now it makes more sense why they work on some bacteria but not others.
Exactly. Penicillin and its relatives work by interfering with the final step of peptidoglycan synthesis, specifically linking the peptide cross bridges together. This weakens the cell wall. It's generally much more effective against gram-positive bacteria because they have that thick, exposed peptidoglycan layer that's constantly being synthesized as the cell grows. Gram-negative bacteria are less susceptible, partly because the outer membrane prevents penicillin from easily reaching the thin peptidoglycan layer, and partly because they have fewer peptide cross bridges to target.
Okay, so we've explored the outer defenses, the cell wall, and how bacteria move. Let's dive deeper inside the cell wall now. What's just beneath that protective layer?
Right under the cell wall, you have the plasma membrane, sometimes called the cytoplasmic membrane. It's a thin, very dynamic structure that encloses the cell's cytoplasm. We describe its structure using the fluid mosaic model. It's essentially a lipid bilayer, primarily made of phospholipids arranged tail to tail, with various proteins embedded within or attached to it. These components aren't fixed. They can move around laterally, hence fluid mosaic. Eukaryotic membranes also have carbohydrates attached and sterols embedded for stability, which most bacterial membranes lack except for those wall-less mycoplasmas we mentioned.
What's its most important job? What does this plasma membrane do for the cell?
Its most critical function is acting as a selectively permeable barrier. It controls precisely what gets into and out of the cell. It's the gatekeeper. Small, uncharged molecules like oxygen, carbon dioxide, and water can often pass through relatively easily by diffusion. But larger molecules, like sugars or amino acids and charged ions, require specific transporter proteins to help them cross. It's not just a barrier, though. It's also where many crucial metabolic reactions happen. In bacteria, since they lack mitochondria, the plasma membrane contains enzymes involved in ATP production through cellular respiration. In photosynthetic bacteria, it has infoldings called chromatophores containing pigments for photosynthesis.
So it's absolutely vital for the cell's life, which means it's also a target, right? What kinds of things can damage it?
Absolutely. Since it's so vital, it's a target for certain antimicrobial agents. Things like alcohols and quaternary ammonium compounds, common disinfectants, disrupt the membrane lipids. And there's a group of antibiotics called polymyxins. These specifically target the phospholipids in the bacterial plasma membrane, especially in gram-negative bacteria. They basically punch holes in it, causing the cell contents to leak out, leading to cell death. This is actually relevant clinically. Sometimes, if you kill gram-negative bacteria too quickly with an antibiotic that breaks the cell wall, you get a massive release of endotoxin, lipid A, which can be harmful. Polymyxins kill by disrupting the membrane, which might sometimes be preferred.
Okay, that makes sense. Let's talk a bit more about how things actually move across this selective barrier. You mentioned transporter proteins.
Right. Materials move across the membrane via two main kinds of processes, based on whether the cell needs to expend energy. First, you have passive processes. These don't require the cell to use ATP energy because substances are moving down their concentration gradient from an area of high concentration to low concentration. The simplest is simple diffusion, where small molecules just move across until they reach equilibrium. Then there's facilitated diffusion, which still moves substances down their gradient, so no energy needed, but it requires a helper protein, either a channel or a carrier protein, to get across the membrane, useful for ions or larger molecules like glucose. And a special case of passive movement is osmosis. This is specifically the net movement of water across a selectively permeable membrane, moving from an area of high water concentration, low solute, to an area of low water concentration, high solute. Osmosis is critically important. It explains why cells are sensitive to the solute concentration of their environment. In an isotonic solution, the concentrations are equal, no net water movement. In a hypertonic solution, like pure water, water rushes into the cell. If the cell wall is weak or absent, like our protoplasts, the cell swells and bursts osmotic lysis. In a hypertonic solution, high salt or sugar, water moves out of the cell, causing the plasma membrane to shrink away from the cell wall. This is called plasmolysis, and it inhibits cell growth. It's the principle behind using salt or sugar to preserve food.
Okay, so that's passive. What about when the cell needs to move something against the gradient?
That requires active processes, which do require the cell to expend energy, usually in the form of ATP. Active transport uses specific transporter proteins, often called pumps, to move substances across the membrane against their concentration gradient from low concentration to high. This allows the cell to accumulate needed nutrients, even if they're scarce outside. Then there's a process pretty much unique to prokaryotes called group translocation. This also requires energy. As a substance is transported across the membrane, it's chemically altered. For example, glucose might have a phosphate group added to it. This modification traps the substance inside the cell because the transporter is specific for the original substance, not the modified one, and it also prepares it for subsequent metabolic pathways. Very efficient.
Okay, so we've got the membrane, the gatekeeper, and how things move in and out. What's filling up this bustling, selectively permeable DAG then? What's inside the cytoplasm?
Right, the cytoplasm. It's the substance inside the plasma membrane. It's mostly water, about 80%, but it's packed with everything else the cell needs to function. It contains proteins, especially enzymes, carbohydrates, lipids, inorganic ions, and low molecular weight compounds. It appears thick and granular because of all the ribosomes in there. Importantly, prokaryotes also have a cytoskeleton, though it's simpler than in eukaryotes. It's made of proteins like MURI-B, FTSZ, and others, and it plays roles in maintaining cell shape, cell division, and maybe even moving DNA around. Unlike eukaryotes, though, prokaryotic cytoplasm generally doesn't exhibit obvious streaming movements.
And the most important molecule of all, the genetic blueprint, the DNA, where is that located in the cytoplasm?
It's concentrated in a region called the nucleoid. This isn't a true nucleus because, remember, there's no membrane surrounding it. The nucleoid typically contains the cell's main genetic material, a single, long, continuous, and usually circularly arranged thread of double-stranded DNA, the bacterial chromosome. This carries all the essential genes for the cell's life. But bacteria can also have additional, smaller, circular, double-stranded DNA molecules called plasmids. These are separate from the main chromosome and replicate independently. Plasmids aren't usually essential for survival under normal conditions, but they often carry advantageous genes, things like genes for antibiotic resistance, tolerance to toxic metals, the production of toxins, or the ability to synthesize certain enzymes. And critically, these plasmids can often be transferred from one bacterium to another through that conjugation process we mentioned earlier. Big implications for adaptation and disease.
Plasmids, right. Key for resistance spread. Okay, so protein synthesis, the reading of that genetic information, where does that happen in this busy environment?
That happens at the ribosomes. Ribosomes are the sites of protein synthesis in all cells. They translate the genetic code carried by messenger RNA into actual proteins. Prokaryotic ribosomes are slightly smaller and less dense than eukaryotic ones. We call them 70S ribosomes, and they're composed of two subunits, a smaller 30S subunit and a larger 50S subunit. The S refers to Svedberg units, a measure of sedimentation rate. They don't simply add up. This difference between prokaryotic 70S ribosomes and eukaryotic 80S ribosomes is absolutely crucial for medicine. Many important antibiotics like streptomycin, gentamicin, erythromycin, and chloramphenicol work by specifically targeting and inhibiting protein synthesis at these bacterial 70S ribosomes while leaving our own 80S ribosomes relatively unaffected. That's the basis for selective toxicity.
Selective toxicity, right, that's the goal. Finally, inside the cytoplasm, they sometimes have little storage units called inclusions.
Yes, inclusions. These are basically reserve deposits found within the cytoplasm of some prokaryotic cells. Cells can accumulate certain nutrients when they're plentiful and then use them later when they're scarce. Storing them as inclusions avoids increasing the osmotic pressure inside the cell too much. There are various types. Metachromatic granules, for example, store inorganic phosphate they stain distinctively and are diagnostic for Corynebacterium diphtheria. Others store polysaccharides like glycogen or starch or lipids. Some bacteria store sulfur granules as an energy reserve. Others have carboxysomes, which contain enzymes needed for carbon dioxide fixation during photosynthesis. Aquatic prokaryotes might have gas vacuoles, which are hollow cavities that maintain buoyancy, allowing them to float at an optimal depth for light or nutrients. And some even have magnetosomes, inclusions of iron oxide that act like tiny magnets, potentially helping them orient themselves in magnetic fields, maybe to find nutrient-rich sediments.
Magnetosomes, that's amazing. Okay, this next one is truly remarkable, though. Some bacteria seem to have an incredible superpower for survival, something called an endospore. What's that about?
You're right, it's one of the most fascinating aspects of microbiology. Endospores are essentially dormant, highly resistant resting cells formed by certain gram-positive bacteria, most famously those in the genera Clostridium and Bacillus when they encounter harsh environmental conditions, especially the depletion of essential nutrients like carbon or nitrogen. These are not reproductive structures, they are survival structures. They're highly dehydrated, have incredibly thick walls, and possess additional protective layers beyond the normal cell wall. This complex structure makes them extraordinarily resistant to extremes that would kill normal vegetative cells, things like high heat, lack of water, toxic chemicals, and even UV radiation.
So they can just wait out bad times
Hmm.
for potentially a very, very long time.
Precisely. They can remain dormant and viable for incredibly long periods, potentially thousands, maybe even millions of years under the right conditions, just waiting. The process of forming an endospore within a vegetative cell is called sporulation or sporogenesis. It's complex. A copy of the chromosome in some cytoplasm gets isolated and surrounded by multiple protective layers, including a thick spore coat. Endospores contain high levels of calcium ions complex with dipiclinic acid, DPA, a chemical unique to spores, which helps to stabilize their DNA and proteins against damage, especially from heat. Then when environmental conditions become favorable again, presence of nutrients, water the endospore undergoes germination. This is the process of returning to the metabolically active, growing vegetative state. And again, it's important to remember this isn't reproduction. One vegetative cell forms one endospore, and that one endospore germinates back into just one vegetative cell. It's purely about survival.
And why is this clinically and industrially so important? What are the implications?
Huge implications. Because of their extreme resistance, endospores are a major challenge in healthcare and the food industry. Standard boiling doesn't reliably kill them. They can survive boiling water for hours. This necessitates much harsher sterilization methods like autoclaving, steam under pressure, to ensure medical equipment is truly sterile. It's also why canning processes for food must be carefully controlled to destroy potential endospores, particularly those of Clostridium botulinum, which causes botulism. Their resistance means that Clostridium species are significant causes of diseases like tetanus, gas gangrene, botulism, and certain types of food poisoning and antibiotic-associated diarrhea. And Bacillus species are known for causing anthrax and certain food poisonings. Their resilience, however, also makes some Bacillus spores useful as biopesticides.
Incredible survival mechanism. Okay, we've done a really deep dive into prokaryotes, particularly bacteria. Now let's spend a little time looking at our own cellular cousins, the eukaryotes, to understand how they compare and maybe touch on how scientists think they evolved.
Right. So eukaryotic cells, again, it's plants, animals, fungi, protozoa, algae, are typically much larger and structurally far more complex than prokaryotes. They also often have projections used for locomotion or for moving substances along the cell surface. Flagella and cilia. Eukaryotic flagella, as present, are usually few and long. Cilia are numerous and short, like tiny oars. Importantly, unlike the rotating propeller-like motion of prokaryotic flagella, eukaryotic flagella and cilia move in a complex, wave-like or beating manner. And internally, they have a very characteristic 9+2 arrangement of microtubules, a complex internal structure completely different from prokaryotic flagella. Now, looking at their outer layers, eukaryotic cell walls, when they're present, like in plants, algae, fungi, are chemically much simpler than bacterial peptidoglycan walls. Plant cells use cellulose, fungi use chitin mostly. Animal cells like ours lack a cell wall entirely. Instead, animal cells are often covered by a glycocalyx, a layer of sticky carbohydrates extending from the plasma membrane. This helps with strengthening the cell surface, attachment to other cells or surfaces, and crucially cell-to-cell recognition. And a critical point of comparison, no eukaryotic cell naturally contains peptidoglycan. This is a cornerstone of antibiotic therapy targeting peptidoglycan synthesis, harms bacteria but doesn't harm our eukaryotic cells.
Right. And the plasma membrane in eukaryotes. Similar functions, but maybe some differences.
Similar basic structure, the fluid mosaic model still applies, and similar function as a selectively permeable barrier. However, eukaryotic plasma membranes contain sterols, primarily cholesterol in animal cells. These molecules fit into the lipid bilayer and help to strengthen the membrane and make it less fluid, especially at higher temperatures. Most prokaryotes lack sterols, mycoplasma being the exception. Eukaryotic membranes also have carbohydrates involved in the glycocalyx for attachment and cell recognition. And importantly, eukaryotic cells can perform endocytosis. This is the process where the plasma membrane folds inward to bring substances into the cell. This includes phagocytosis, cell eating, for taking in large particles or even whole cells, and pinocytosis, cell drinking, for taking in fluids and dissolved molecules. Bacteria generally don't do endocytosis.
Okay, inside the eukaryotic cell now, the cytoplasm and ribosomes.
The eukaryotic cytoplasm encompasses everything inside the plasma membrane and outside the nucleus. Like prokaryotes, it has a cytoskeleton. But the eukaryotic cytoskeleton is much more complex and extensive, composed of microfilaments, intermediate filaments, and microtubules. It provides support, shape, and allows for movement of organelles within the cell, and even cell movement itself. Eukaryotic cytoplasm also exhibits cytoplasmic streaming, the movement of cytoplasm throughout the cell, which helps distribute nutrients and move organelles around. We don't really see that in prokaryotes. As for ribosomes, eukaryotic ribosomes located in the cytoplasm and attached to the ER are larger ADS ribosomes, composed of 60S and 40S subunits. But here's that curious detail again. The ribosomes found within the mitochondria and chloroplasts of eukaryotic cells are actually 70S ribosomes, just like those found in bacteria. Keep that in mind.
70S ribosomes inside eukaryotic organelles. Interesting.
And of course, the defining feature of eukaryotes, the organelles themselves. Can you give us a quick tour? Absolutely. The compartmentalization is key. You have the nucleus, contains the cell's chromosomes, DNA complex with histone proteins, enclosed within a double membrane called the nuclear envelope, which has pores to regulate traffic. It's the control center. Contains the nucleolus, where ribosomal RNA is made. The endoplasmic reticulum, ER, an extensive network of membranes continuous with the nuclear envelope. Rough ER has ribosomes attached and is involved in synthesizing proteins destined for secretion or insertion into membranes. Smooth ER lacks ribosomes and is involved in lipid synthesis, detoxification, and calcium storage, the Golgi complex. Stacks of flattened membrane sacs. It receives proteins and lipids from the ER, modifies them, sorts them, and packages them into vesicles for delivery to other organelles or for secretion out of the cell, like the cell's post office. Lysosomes, vesicles formed from the Golgi that contain powerful digestive enzymes that break down worn-out organelles, ingested materials, and cellular debris. Vacuoles, membrane-bound sacs that can have various functions, storage of water, nutrients, or waste products. Plant cells often have a large central vacuole important for turgor pressure. Mitochondria, the powerhouses. These are the primary sites of ATP production through cellular respiration. They have two membranes, the inner one folded into cristae. They contain their own circular DNA and those 70S ribosomes, and they reproduce somewhat independently within the cell by binary fission. Chloroplasts, found in plant cells and algae. These are the sites of photosynthesis, containing chlorophyll pigments. Like mitochondria, they have inner and outer membranes, their own circular DNA, 70S ribosomes, and reproduce by binary fission. Peroxisomes, small organelles containing enzymes that oxidize various organic substances, often producing hydrogen peroxide as a byproduct, but they also contain catalase to break down that toxic peroxide. And the centrosome, located near the nucleus, it's the main organizing center for microtubules and the mitotic spindle during cell division in animal cells.
That tour really highlights the complexity, but it circles back to that question you raised earlier. Why do mitochondria and chloroplasts have so many features, their own circular DNA, 70S ribosomes, binary fission reproduction that seems so, well, prokaryotic?
Exactly. It's striking, isn't it? This observation is the cornerstone of the endosymbiotic theory, which is the leading scientific explanation for the origin of eukaryotic cells. The theory proposes that eukaryotic cells evolved from a symbiotic relationship that began when a larger ancestral prokaryotic cell, perhaps an early archaeon, engulfed smaller prokaryotic cells. Instead of being digested, these engulfed cells survived inside the host. Over millions of years, this turned into an obligatory symbiosis. The host cell provided protection and nutrients, while the engulfed cell specialized. Those that could perform aerobic respiration efficiently became mitochondria. Those that could perform photosynthesis became chloroplasts. They became integrated organelles rather than separate organisms.
And the evidence for this seems quite strong.
Very strong. The points we've mentioned are key evidence. Mitochondria and chloroplasts are similar in size and shape to bacteria. They contain their own circular DNA, lacking histones, just like typical bacterial chromosomes. They have 70S ribosomes, which are characteristic of prokaryotes and sensitive to the same antibiotics that target bacterial ribosomes. They reproduce within the eukaryotic cell by binary fission, independent of the host cell's nuclear division, mitosis. And their inner membranes have enzymes and transport systems similar to those found in the plasma membranes of bacteria. It all points very strongly towards an ancient endosymbiotic origin.
That's a fantastic explanation of how complexity might have arisen. So there you have it, a really whirlwind deep dive into the incredible functional anatomy of cells. We've journeyed from those fundamental distinctions between prokaryotes and eukaryotes right through to the intricate roles of cell walls, membranes, ribosomes, and all that internal machinery. And we've seen how these tiny structures directly impact everything from disease and antibiotics to the very evolution of life itself.
Absolutely. Understanding these foundational differences isn't just, academic textbook stuff. It really unlocks why certain medicines work and others don't, how infections spread and sometimes resist treatment, and the truly ingenious ways microbes adapt and survive in almost every environment imaginable.
Thank you so much for joining us on this deep dive. We really hope you've gained some fascinating insights and maybe had a few aha moments about this microscopic world that's all around us and inside us.
Definitely. Until next time, keep exploring the microscopic wonders around and within you. There's always more to learn.
And as always, we'd like to leave you with a provocative thought to mull over. Considering the incredible adaptability and sheer survival power of prokaryotes, especially things like endospores that can survive for ages, and their ability to form those incredibly resilient biofilms, what does this really tell us about the ultimate limits of life here on Earth, and perhaps maybe even beyond? Something to think about.